genomics of Ebola, HIV, and COVID-19 in Africa. And as Deus has said, I had the World Bank African Center of Excellence for Genomics of Infectious Disease at Redeemers University. So we are a center that is, we are at the WHO and Africa Union, reference laboratory for genomics and research and innovation, in Africa. And as such, we have a mentoring role and then also quality assurance and supervisory role over the national and the regional labs across the 54 African countries. So today I will probably just go, and then as we talk about, we're in the era of pandemic, but then, on these slides is basically to show the position of Africa. And if you see, for instance, in this map, and then you look at all of these viruses, and these viruses that have pandemic potential, which includes HIV, Ebola, SARS-CoV-2 now, you realize that the majority of them are in Africa. And then the reason, why Africa, and then that really points to the fact that naturally, Africa should have been the world leader in terms of preferring solution to this, to this disease, or to these pathogens, that have pandemic potential across the globe. So across those, among those pandemic, you have Ebola, you have Lassa, you have HIV, you have COVID-19, although did not originate or emerge from Africa. But then this is really one clear evidence why, Africa should be the leader, in this area. So we really were very cognizant of this fact. And then as far back as 2010, we started thinking about how do we give Africa the prominence that it needs and the place that it deserves in a community of nations or in the globe, because Africa is endowed not only with mineral resources, but also with very extensive biodiversity. And as part of that biodiversity are viruses and pathogens. And naturally, Africa should be the world leaders, as I said before. So we thought about, how we can start bringing in genomics, in the early 2000s, in this space, simply because we realized, that genomic sequencing technologies have revolutionized the way healthcare research and research has been approached. And that has created a huge divide between Africa and the rest of the world. And these divides are not only financial, but also in terms of, benefit in enhancing science. And we thought that, embracing genomics and then start bringing that genomic revolution on the continent to address things that are very peculiar to Africa would have actually been very tremendous. And in the next few slides, we will see how we really started pioneering this on the African continent and how we're able to use that as a way to transform things like outbreak response to diseases. And I think to do that, we set up a platform, and then that platform, the African Center of Excellence for Genomics of Infectious Disease. And on this slide, you see, the African countries that are involved in this, and then also the partners, overseas partners, even though this has grown so much. So, and the mission for the platform was really to create a vibrant academic and research environment that transcends national boundaries, where we can ensure, proactive, responsive, and proactive and ethical, high quality translational genomic research in Africa, with the focus in this case of infectious disease. There are also that are working in the space of non-communicable diseases. So, and then our goal was really to start thinking about was to develop that critical mass of well-trained African scientists that can use, that can be empowered, to use genomic-based tools and knowledge in order to work toward prevention and elimination of infectious diseases. And in order to do that, for instance, we thought about not only to provide short-term training for specialized skills, but also thinking about long-term training, that is training those who could be trainers of two orders. And then as such, we set up an international, master's and PhD program that, would focus on genomics, and infectious disease. And also hoping we are going to be training people how to engage communities for public health, action. So, and the research driving this program actually was focused around something that is very peculiar, that cut across most infectious diseases on the continent, that is actually identifying, the pathogens that are driving febrile illness on the continent. And that we could actually not only unravel, what we call the microbial landscape, but actually using such information as a way to provide, solutions. Solution, we're talking here in terms of diagnostics, we're talking about therapeutics, and then we're talking about vaccines. And that's really what we've tried to do over the past few years. So in bringing this, this education platform and the research platform and the research platform together, we realized that we're really meeting one of Africa's critical needs for genomic education and research. And then that really, we should look at this Venn diagram, that where the education and research cut across is really about genomic capacity building. That is building infrastructure, training, and also setting up diagnostic core facility, and also more importantly, actually providing, a platform or providing, the leeway for sustainable, careers development on the continent. And ultimately, our goal is basically not just to do the research and then the academia work that we're doing, but is actually to build a next generation of African pathogen hunters who will hunt this pathogen, characterize them, and then prefer solution in Africa, and then therefore provide and giving Africa this prominent role that it actually deserves, in the community of nations, and then give Africa a voice, when people are talking about providing solution to problems. So, and then one of the very first demonstration for this really came out of the 2014, 2014 to 2016 Ebola outbreak in West Africa, where we, for the first time, actually demonstrated the transforming power of genomics in terms of, guiding outbreak response. And this was the first time this was being done. And then I know we faced a lot of challenges at the time where we were talking about how do we use genomics and then create, you create open data sharing or open data access, in order to actually use information and make information actionable in order to guide public health response. And we're glad that, what we pioneered at the time in 2014 has become the standard. Today, we're hearing about GISED and people dropping, data in GISED, all of the countries in the world. But yet, the story is not being told completely. We pioneered that whole process in 2014 by making available the first 99 genomic sequences on the continent. Became very important because at the time you really did what the world did not thought that it was impossible to do. As I was saying before, we generated the first 99 genome of Ebola. We made it open, accessible. So which means we pioneer open data sharing and open data access for vaccine developers, for clinical, for therapeutic developers, and then for diagnostic developers. But then what is really important is the fact that we came up with a new way and a new technology that is basically transforming, the way you could even do genomic during an outbreak. A process that could have taken about a month, we actually cut down, developed new protocols, and then made it within 10 days. That is from sample collection to generating, the sequence data and then making the data assembly and then dropping them in NCBI. This was really 10 days. And this is something that was never seen before. So, and this is what we call transformation. We made this information available for months, and then our publication really could not come out about 6 months after we dropped the information, opened the data, to the international community. So, but what is really important I want to show here is actually the power behind genomic, where we show for the first time what is being used today as a standard. That is the case of Sierra Leone, where we show the incursion of 3 different lineages in the country, follow that over time, and then we show that, at some point around June, for instance, one single lineage has taken over and became the most transmissible lineage, and this was the first time you could use this kind of, use this kind of platform to demonstrate, the evolution, of how you could have new variants taking over in the process of an outbreak, showing that, within an environment or within a country, for instance, we had, sustained human-to-human transmission. There was no introduction of new variant from animal reservoirs, and there was no imported, it was that community transmission going on in the country. And then this information were given in real time to public health authorities. For the first time also, that actually we identify some specific, within the specific areas of the genome of those viruses, and then we actually characterize this particular variant that was associated with increased transmissibility. So, and this has become really the standard, and then, and then we really have, we feel happy that we actually set a standard in 2012. But what is really critical here that I want to show you in this slide is how, for the first time in the history of science and then in epidemiology, we're able to identify those viral genome factors or virus genome factors that actually that encourages or sustain the spread of Ebola within not only Sierra Leone but across West Africa except Nigeria. So if you look in this slide, as you see in times and then in space, you could see here how we're showing within time and space how the virus is transmitting from person to person, country to country, countries to countries, and then showing the how, and this really actually was a very first demonstration of genomic epidemiology in action, and this information became very critical, in terms of containing this outbreak. But what we've done, as I've mentioned before, that is translating genomic into what we call point-of-care diagnostic. We started with high-fidelity sequencing. Yes, we actually, also use that to refine the PCR diagnostic, but we realized, for instance, that PCR was taking a long time. And what you need during an outbreak, are two factors: speed and accuracy. Speed of the process, speed of execution, and then accuracy, with what you're doing. And that's what we did here, basically by going from genomic data and translating that into what we call the very first rapid diagnostic test that could detect Ebola and confirm Ebola in 10 minutes. And this was the very first rapid diagnostic test that was approved by the WHO and then the US FDA. And then you could see that here. So the introduction of this test in West Africa was one of those major things that started bending or flattening the curve, because the doctors or the people could see in real time what Ebola was all about. So we actually applied the same knowledge and same skill almost in real time in Nigeria. And that's how Nigeria got in the history, the annals of medicine, and the annals of epidemiology, because this country of more than 200 million people, we actually leveraged knowledge, skill, and genomic technology in order to guide the government in its public health response. And then Nigeria actually ended up with only 20 cases and 12 deaths. But this was really genomic and epidemiology in real time, where we could identify the areas of hotspot and guide the government. So this is really showing one example how you could actually use genomic power in order to respond to an outbreak. So again, the second thing that I want to mention here is actually to show the power behind genomic and infectious disease. This is a case of a virus called Lassa fever, Lassa fever, that emerged in Nigeria, or that was described for the first time in Nigeria about 53 years ago, and then this virus was circulating in this country, and then for decades, there was no diagnostics. And then the sample used to be sent to Germany, and then often when the result of the sample come back, then 90% of the people or the cases are dead, and then these results were completely useless. In 2007, I took that challenge actually to see how we could explore and then develop diagnostics for this virus. And then within 6 months, we set up a next generation platform. By 2011, we went on to sequence viruses directly from patient samples and rodent samples, and effectively, came up with the first genomic map of the Lassa fever virus. Not only we set up the map, but we also, made some very interesting findings. One, for instance, is that this virus that was described in Nigeria just about 53 years ago, we found that virus using the most remote common ancestors is actually about 1,060 years ago, and it emerged from present-day Nigeria. It was circulating within Nigeria years and moved out of Nigeria about 400 years ago. Yeah, what is really important about this finding is the fact that we not only created the largest genome data set for the Lassa virus, but we also translated that knowledge in developing the pan-Lassa rapid diagnostic test, and this has been very critical for the control of this disease, over time. But what are the lessons here, and then really the message here behind this genome, the whole genome sequence of the Lassa virus, is the fact that in the past 5 years, between 2015, when we published this sequence data, and now there have been 3 major vaccine candidates that have been developed from the sequence data that we actually generated. That is showing how you could speed up a process and then find solution to a problem that has been ignored over the years. So, and then that really is very helpful in the sense that now we have 3 vaccine candidates against a disease that was neglected over decades. So this, we really were lucky to just make this publication, but also had the cell coverage. And then again, here is showing again powerful genomics that we did the first metagenomic clinical samples of pathogen, in Africa. And then this is here to show the power behind pathogen discovery. So using metagenomic as a way to go on and discover 2 novel rhabdoviruses, and then name those viruses Epomavirus 1, AKV1, and AKV2 from the area where we described. So what is critical about this, or what is important about the 2 new viruses, is the fact that they actually have a strong similarity with the Bakongo virus, a virus that was discovered in the DRC and was responsible for a viral hemorrhagic fever over there in 2012. But interestingly, in Nigeria, for instance, when we went back and then looked into those people, they all had like very high exposure to this virus. Then, Genomics for Public Health actually having demonstrated that in 2012 or 2014 with Ebola, we actually had to use that again in 2018 when Nigeria was faced with a major surge of the Lassa fever virus. There were many, public health questions that were asked, when we're dealing with new variant. It was a surge due to human-to-human transmission. You're talking about 200-fold increase within 8 weeks. So that is, you have more cases in 8 weeks than you've had in the past 2 years combined. So then this question, we actually to swing, we use genomic as a way in real time to respond to this question, and then to help the WHO and the Nigeria Center for Disease Control to address this problem. We sequence these viruses, and then we made information available. Thanks to the guys in my lab, then we demonstrated that there was no evidence of particular new strain or no evidence of increased sustained human-to-human transmission, but that this was actually, the search was due to multiple introduction of genetically independent viruses. Depending on where you were in Nigeria, you could actually see that the northern part of the virus, Nigeria has a variant of 3 or lineage 3, the west was lineage 2A, and then the east was 2B. So basically a country dealing with an outbreak in 3 dimensions, depending on where you were with the virus. So this was a shout out by WHO and then Africa Cities and the Nigeria Center for Disease Control for what we've done. But again, although we published this paper in New England, we realized that there was one major caveat. That caveat was actually due to the fact that we realized that the viruses were coming from rodent, and then that really helped, how to link up. We started a nationwide, rodent trapping, and then are trying to look at genetic diversity in the rodent and in human. And for the first time, you could see here in red, we were demonstrating the link between a rodent sample and an animal sample, that is showing the direct link between what happens, in animals and how this is done from animal to human. So, but when we didn't stop there, we actually adapted and updated our games and basically started using, gene editing technology, which is a Sherlock, CRISPR-based technology, to actually develop a novel kind of diagnostics for Lassa fever. So in this case, using Cas13a, and then we're able to actually come up with a diagnostic that could be done at room temperature within 5 minutes. You could get a result, and that really became very important. That is basically describing the whole process here, where you use your Cas guide RNA and then adding on the nucleases and then adding a reporter, so you end up having a diagnostics like the one that I'm showing here. So, and then this really also became like a major breakthrough. There was the first demonstration of using CRISPR-Cas13a, for instance, for a disease, and then this really showing the team in my lab. Doing all this work. And you can see here one of my graduate students, Jessica, Victoria, doing this work. And then this was really a nice publication into Nature, where Amy Maxson embedded in a lab for a week actually to see this work being done from the beginning to the end of the process. So this other slide is really actually to show also real-time genomic analysis, when you want to talk about how you can use this actually to uncover outbreaks. So in this case, this is a case where we use metagenomic to uncover an outbreak of yellow fever somewhere in the southern part of Nigeria. So this was in 2018. We had, within a week, we had 179 school children that died in the community. All the tests, proved negative. Lassa, Ebola, and everything proved negative. The Nigeria Center for Disease Control did not understand what was going on. They sent us 22 samples, and then we went in there with very powerful metagenomic approaches, and we were able to identify a new lineage. So here, new strain, you could see them clustered in red, of yellow fever circulating in Nigeria. But what is important about this finding is the fact that this new strain of Lassa fever, yellow fever, that was circulating in Nigeria and was negative for all the tests, was due to the fact that it was diverse and it was very different from the ones that have been circulating in the country in the past 94 years. So really showing how if you embed, like this kind of powerful technology in the silos, laboratory silos method that are existing today, you can make a big difference. And then this is again a typical case also whereby in Nigeria, this paper came out about on Monday this week, where we're able to use, next generation sequencing to identify an extensively drug-resistant tuberculosis in Nigeria and West Africa. And this is the very first time, that this description is made in West Africa, whereby we really use genomic data actually to prove that we're dealing with a strain of TB that is not responding to any of the drugs that are available. So really showing the again, behind the detection of this technology, so we can actually show clearly. So the case of HIV, for instance, is another case here, for instance, where we use, genomics as a way. I mean, one of the first, whole genome sequence project being done in Africa, where we were actually not just looking at the envelope or the GAG or the pole gene of HIV to characterize the HIV-1, but really looking at whole genome sequence. And that has really helped a lot, because it not only just helping us to understand exactly the genome, the HIV diversity, but also understanding actually spread and an importation of diseases. In this case, for instance, we've been able to show how the viruses have been brought into Nigeria from neighboring countries by just using genomic sequences to track, some of these viruses migrating or moving or migrating through human from country to countries. And that becomes very important as a way, to do this. So again, just along the line, we have a very powerful bioinformatics team that is developing new toolkits. For instance, like here, this is something that is published, will be published on Monday in PeerJ, which is a bioinformatics journal. This is basically a viral genomic, assembly platform that my team has just assembled, and then you don't need a server, you don't need, just download it on your system or your laptop, and then you can actually do all of the bioinformatic analysis, just put in your data, and then you can get the output. Now, let's fast forward to COVID, and then COVID, as you know, really struck Africa, a bit early in 2020. And then the very first country that actually had COVID, one of the first countries was Nigeria. And then when we got the samples in Nigeria, when the first suspected case arrived in Nigeria, then the Nigeria Center for Disease Control had to just get the sample down to us, to our lab, basically knowing exactly the capacity that is available and then the level of automation that we have here. So you could see here, for instance, the automation and then the platforms, and then you could see here the next 6,000 still remain the only piece in Africa. So, but then we basically just pushed this, and then within 48 hours, we're able to actually first, come up with the first SARS-CoV-2 genome sequence, in Africa. But then this information became very critical, because we knew that definitely this information, this virus was imported and it wasn't circulating in Africa. And then together with other colleagues, in the next few months that followed, we were able to show, for instance, that there was about 761 importation of the virus within the continent. And then these were mostly, this data was shared with the Africa Center for Disease Control, and the reason why Africa shut down during the first and second wave at the same time, actually preventing the continent to go to spiral down like other part of the world. But then we use this information not only to inform public health policy, but also to set up a surveillance system within Nigeria. So, but then this slide is a bit outdated. I think over the years, the past over 1 year, we've done over 1,000 sequences. And we've covered more than 2/3 of the Nigerian states. But then in the case of Nigeria, we know exactly at each level where, in each state, what are the variants circulating, and these informations are given almost in real time to the National Center for Disease Control and the Presidential Task Force, so that they can use in order to respond to the pandemic. So in this case, for instance, this is an next-strain analysis. So then, and this information is shared, with the Presidential Task Force for policy making. So a lot of the decisions that they're making countries are based on this data. So which means that the information or the policies are evidence driven. We know the different lineages circulating in Nigeria. In the last count, there were over 45 different lineages. So we know exactly during the lockdown, even in Nigeria, we know, if you look at this year between the different states, you could see variant moving from state to from state to state. And that's really was very helpful at the time to help the government in order to devise strategies, to prevent, people to move, because we realized at some point, despite the lockdown, things were moving. So, and one thing I want to highlight here is the fact that, the genome sequence data that we generated were not just used for public health, response, but we also translated that into point of care diagnostics here. This is all a SHINE technology that we've just developed. This is also done at room temperature. And then you have the format that could be done on strips that can be done in the most remote part of the world, but we also have the plate format that can be done, to increase the throughput, where we could do 96 samples at a time. And then this kind of a very first test that we did in Nigeria. At the time in our lab, like 500 first tests, and that was very happy. Happy to see that. But then what is very important is the fact that this was within 2 weeks of the outbreak, and that we're able to come up with this new technology out there. But what is important in the fact is the fact that we know now that within a week, if we have an outbreak, we should be able to come up with a solution and then help, help with this. But interestingly, we are also now multiplexing, this in our lab, and basically bringing together all of this technology, all of this pathogen under one single diagnostic chip, and whereby we could actually go, test up to over 160 different pathogens in one single test and assay. We've also translated this into coming up with like a vaccine candidate for some of you that know, and that has gone through preclinical trials, and it will moving on to clinical trials. But the story behind the vaccine and the reason why we're not funded to compete with others, because it's again the same messenger RNA technology platform that we developed and designed here, but we couldn't move it forward because of science politics and also lack of support from African government. So I really would not want to bother you with all of this, but then this is a subject to a different conversation. But one thing that we basically want to say here is that actually we basically designed a vaccine candidate that is mostly adapted for the continent. We do not need temperature change. We do not need, a needle. We definitely is a needle free because we understand exactly the context in which we evolve in. So, and then again, to say that over time, we've not just worked only with Nigeria, we've also supported many African countries. And as I can see here, identifying variants of concern and variants of interest. So we've basically done a lot in supporting not only Nigeria, but many African countries. And then this is here, for instance, whereby we've been able to help about 14 different African countries to sequence, their samples, and that has generated over 2,500 different genome sequences of SARS-CoV-2. Training also the capacity in those different countries has helped. And then in this case, for instance, we've generated over 2,500. We've also trained a critical mass of people since 2014, over 1,000. But what is very impactful is the fact that the case of countries like Ghana, for instance, Brigadu Noguchi is someone that we train, and then today is one of the leaders in West Africa in performing genomic sequences. And then we also have the case of Rwanda that we actually trained Rwandese scientists, and then we went back to their countries, and within a week they started generating sequences on their own. And that has been very, very helpful because we actually building that capacity on the continent in a way that was unprecedented. But what we've done is that and what we've been doing is not just to train, but we're also following that through with what we call infrastructure. We're setting up sequence platform across the continent, and that really has been very helpful in terms of helping Africa to respond. Finally, what I'm going to close on is actually to talk about this new platform called Sentinel, a new project called Sentinel that is really built around making Africa lead pandemic preemption and response, and build on genomic technology and then what I call information technology as well, and information sciences. It's really built around three major pillars. The first is to detect known and unknown infection almost in real time, and then pass that information to public health authorities, locally, nationally, and then internationally. That brings us to the second pillar called Connect. And then the final one is empower. How do we empower the public health community, including, the national public health and then the frontline healthcare workers, in order to harness these tools and technologies in order to respond to the pandemic on time? You all agree with me that if we have this kind of platform set up before, I'm sure this would have been detected, or the SARS-CoV-2 would have been detected much more earlier in Wuhan or in China. This information would have been shared globally, and then the world would have responded more quickly. So this would have put us in a very different position compared to where we are now. So our goal is actually to use this system where we build around what we call the hubs of excellence here in orange, and then the spokes, which are the satellite sides of the hub, will now work with the communities. So, and then this is a hub and spoke approach that actually connect the hub and the spokes and then the satellite communities, and so that we can have a global system, or a system that can enable us to respond almost in real time. And then once you have created a system that will connect all of them, and then eventually you will have to empower them by just ensuring that everybody becomes a sentinel, and then we'll start looking. We've started pioneering this system in Nigeria, and then we're actually going to be establishing the same system in Senegal, Sierra Leone, and then the DRC. Yes, we are basically going down to a point whereby we think that we can work with the public health authorities, and then we can actually set up this platform whereby we can actually refine the high functioning technology, working with the communities in order to come up with a situation whereby we can empower, Africa's scientific capability, and also and set up really what we call independent surveillance and outbreak response on the continent. And this model can be replicated across the world. I really want to end by acknowledging, not the team working with me, but also a lot of people funding all of the work that I talked about. And I also want to thank you again, and then for listening. Thank you.